So let's talk about protein synthesis. Protein synthesis occurs after transcription, and it happens as the translation of nucleic acid codes are turned into amino acids that synthesize new polypeptide chains. This will be our DNA template strand, and this will be the DNA non-template strand, or coding strand. Now we'll be talking about how our genes are expressed. This is referred to gene expression, which is known as the synthesis of a protein as a result of the transfer of genetic information from DNA to RNA to protein. A gene is the basic unit of heredity that determines a genetic trait. It is a specific sequence of DNA that encodes for proteins and RNA molecules and can contain sequences that influence the production of these molecules. DNA contains the genetic information of an organism, and this information is interpreted or decoded into particular amino acid sequences of proteins. In DNA, the nitrogenous bases are adenine, guanine, cytosine, and thymine, whereas in RNA, the thymine is replaced by uracil. Codons are three nucleotide base sequences that are put together to form one unit in the genetic code of a DNA or RNA molecule. It is a triplet of nucleotide bases in mRNA that is base paired with its complementary anticodon of a tRNA molecule. The promoter codon AUG acts as a start codon, which is a triplet of three bases that specifies the first amino acid of the protein, whereas its stop or termination codon UAA, UAG, UGA sends a signal that indicates the end of the translation of a protein. Transcription is the synthesis of mRNA based on the DNA template of a gene. Messenger RNA, or mRNA, is made during transcription within the nucleus. It contains the genetic information that determines the amino acid sequence of a protein and is used to carry the genetic code from the DNA to the ribosomes. As mentioned earlier, the first stage is transcription, and it happens in three stages. Initiation is the first stage or starting point of transcription and translation in protein synthesis. Elongation would be the cyclic progression of the growth of a polypeptide chain from mRNA that is attached to ribosomes. And termination is the end stage, where mRNA synthesis is put to a stop in transcription. Initiation in transcription. The template strand is the only strand in DNA that is transcribed, while the coding strand has the same sequence as the product mRNA, except the thymine is replaced with uracil. RNA polymerase is an enzyme that catalyzes the synthesis of RNA and binds to the specific promoter region on the gene that is rich in adenines and thymines, which only have two hydrogen bonds and are easier to break. RNA polymerase binds the DNA molecule in which it unwinds a section of the double helix of DNA, reading from template strand in the 3' to 5' direction. In elongation, the synthesis of mRNA complementary to the template DNA is done and the thymine is replaced with uracil. RNA polymerase synthesizes the mRNA in the 5' to 3' direction, just like DNA, but however, it synthesizes only one strand of the DNA template, so no Okazaki fragments are produced. In termination, the RNA polymerase reaches a termination sequence, a stop codon on the DNA strand, UAA, UAG, or UGA, then detaches from the DNA strand, ending transcription. A new mRNA strand is released, and DNA reforms into its double helix. After transcription is finished, modification happens in which the precursor mRNA, pre-mRNA, is converted to mature mRNA. This process starts by the addition of the 5' cap, which is added to the 5' end of the RNA. The 5' cap is a modified form of a gene nucleotide. It allows ribosomes to attach for translation in protein synthesis. Then the addition of the 3' poly-A tail, which involves covalent linkage of a series of A nucleotides to the 3' end of the pre-mRNA, acts as protection against enzymes in the cytoplasm, making the mRNA stable and exist longer, and therefore preventing degradation. The introns which are found in pre-mRNA are then removed, and the addition of exons occurs. We'll be moving on to translation. Translation is the second stage of gene expression that involves translating the nucleic acid code of mRNA into the amino acid code of the protein. The major molecules involved in this crucial stage of protein synthesis transfer RNA, tRNA, and the ribosomes. The first molecule that plays a significant role in translation is tRNA. tRNA is composed of a single strand of RNA and folds into a 2D clover shape. This molecule appears to have 3 loops and a single-stranded region. One end of the molecule is known as the anticodon loop, which is a 3 nucleotide sequence complementary to a specific mRNA codon. The amino acid is a molecule made up of an amino group, acidic carboxylic group, a hydrogen atom, and an organic side group attached to the central carbon atom. It is a building block of protein. in which each amino acid is specifically coded by a codon and linked together by peptide bonds. The second major molecule that plays an important role in translation are ribosomes. Ribosomes are composed of proteins and rRNAs, ribosomal RNAs, that are bound to proteins. They provide a place where mRNA, tRNAs carrying amino acids, and enzymes involved in protein synthesis can assemble and interact with each other. There are two subunits that sandwich RNA, which is the large ribosome unit at the top and the small ribosome unit at the bottom. The P site of the ribosome stands for peptide and is where MET bonds to from the start codon AUG. The A site stands for amino acid, where the new tRNAs bond with the next amino acid that has to be added to the polypeptide chain. The E site stands for exit, and this is the site where tRNA exits. The first stage of translation is the initiation stage, where initiation factor proteins assemble the small ribosome subunit, mRNA. Initiator tRNA, small ribosome subunit attaches to mRNA near the start codon, AUG. An anticodon loop is found on the end of the tRNA molecule, which matches with an mRNA codon. During translation, the anticodon's triplet of bases recognizes and pairs up its bases with a complementary codon found on mRNA. These nucleotide sequences are read in the 5' to 3' direction, while anticodons are read in the 3' to 5' direction. The second stage of translation is the elongation stage, where most of protein synthesis takes place. Once the initiator tRNA is bound to the ribosome, a tRNA with an attached polypeptide is in the P site. A tRNA carrying the next amino acid enters the A site. Then the peptide bond will form between the first and second amino acid and result in a dipeptide that attaches to the amino acid of the tRNA at the A site. Polypeptide is a polymer that is made up of numerous amino acids that are covalently linked together by covalent peptide bonds. mRNA moves along by one codon, and this complex, the polypeptide-bearing tRNA complex, is now at the P site. The uncharged tRNA with no amino acid attached exits the A site. The new codon is at the A site and is ready to receive the next complementary tRNA carrying the next amino acid of the polypeptide. This process continues on with the polypeptide trailing in the cytoplasm of the cell. The last stage of translation is termination, and this is where the stop codon, UGA, on mRNA is reached. The release factor protein recognizes the stop codon and cuts the polypeptide from the last tRNA, helping the ribosome subunits fall off mRNA. The polypeptide is finally released and folds back into a 3D shape. Now this concludes the end of our protein synthesis. As our final protein product, we have the polypeptide with amino acids MET, LYS for lysine, PHE for phenylalanine, and ARG for arginine, and lastly the stop codon. Thank you for listening.
Check this out. I'm very